themselves within action potentials cannot be stronger or weaker. The only thing that's happening is either a signal is sent with the action potential or it is not sent. The intensity, the way that the body measures the strength of the stimulus, because obviously it has to measure whether or not a stimulus is a strong stimulus or not, is not by the strength of the signal, but by the frequency of the signal. So the action potential, if it reaches the threshold, it's going to send the signal along. If the threshold is still there, it sends another action potential along. If it's still there, it sends another action potential along. So with very strong input, with very strong stimuli, action potentials increase in their frequency. Action potentials are sent along very rapidly, very quickly with strong signals, and they're sent along infrequently, very slowly with weak signals. So the way that you sort of measure the strength of a signal with the action potential isn't how strong the signal in itself is, but how rapidly, how frequently the signal is being sent. So pull out your clickers for a second. Action potentials are measured by their frequency, but we mentioned that that's not how gradient potentials are measured. How do you think gradient potentials are measured? Are they actually measured by frequency? Is this a trick question? If the frequency of the signal is the same as the action potential? Are they measured by strength since they're cumulative and they can build an effect? You have the small signals adding up. Can they not be measured at all because they're an entirely different kind of potential? They're not, action potentials can be measured, but gradient potentials can't. Or since it's a continuous signal where signals are always being sent, that it means that they cannot be measured. Think it over, and I realized I forgot to start the clickers, so let me think it over while I do that, and then we'll get going.
And there are two types of the same
No sources.
So I think they're cumulative. I would think. Okay, well, let's say why they're not A. Let's say why they're not all the other numbers on this. So they're not A because you measure it, you add it, it's cumulative. That means it can be strong or weak. What's an example? I'm using graded as my basis for it. Because they're continuous signals, so they can't be measured. It's a type of potential, so I think it's two different types of the same potential. So yes, they can. There are different kind of potentials, so they can't measure the stuff. They're the same type of the certain potential, the nerve impulse potential. Two types of the same.
Yeah, I get it.
Frequency is the same as action. No, they're not the same. There's action and all or nothing. And you can't measure the same thing all or nothing rather than continuous. Because you can measure the same way as long as it's there. Yeah, exactly.
I agree with your
analysis.
Ten seconds left.
Just kidding.
Let's see what everyone said.
What?
That's a pretty overwhelming response there. That is the correct answer. So the thing to remember when you're talking about measuring, anything can be measured if it's producing a tangible result. With the signals that are being sent, what is the measurement that we can do? We can measure the charge. So we therefore know that it's not going to be D and it's not going to be C. Doesn't matter what kind of potential it is, doesn't matter if the signals are being sent continuously or not, we can measure the change in charge. And so that is, we can always measure a result. So the real question is, are they being measured the same way as action potentials, or they'd be measured differently. But with the action potentials, as we said, the strength doesn't matter because it's simply the speed at which the signals are being sent. It's an all or nothing response. If there's a signal there, then we have the stimulus. And it's the speed, the rate at which these stimuli are being sent, the signals are being sent, that we measure the strength. So when we talked about the graded potentials, we said that there are small transient effects that can be cumulative. So they can add up with each other. So that means when you have a stronger signal, you're going to have a stronger stimulus, you're going to have a stronger signal. So you measure it by the actual strength of the signal itself. These signals can add up to each other. More of the transient signals is going to produce a stronger potential. So when we have these transmissions that are occurring, speed is going to be of the essence. You don't want to be able to do something where you stick your hand in a fire and then it takes 10 minutes for that signal to get to your brain, back to your brain to process, send it back and say, you need to remove your hand from the fire. You're pretty much cooked by that point. So you always want to try and make sure that you can send these signals as fast as absolutely possible. Now, anytime you do anything, you can realize how fast the nervous system works. If you don't believe me, look at your neighbor and pinch their arm and see how quickly they respond. So the signals do work fast, but there obviously is a speed factor. You can measure these signals. So if there are ways that you can increase the speed, you can make the nervous system more efficient. And there are two primary mechanisms that the nervous system uses to increase the speed of conduction, to increase the speed of the signal. The first of these is by having axons with larger diameters. If you have a larger diameter axon, remember the axon is the sheet that's moving away from the nerve cell, away from the cell body, transmitting the signal to the next cell, that's going to send the signal faster. So the larger diameter axon you have, the faster the signal can be, and so the cells try and make these axons as large as possible. Basically, the reason for this is because you have less resistance. Anyone here ever take any electrical engineering or anything like that? One person. So you know that resistance decreases with diameter. It's basically a principle of electricity. So if you have a large diameter, you have less resistance to the flow, the current can move faster through the axon. So this is a mechanism that's primarily used in invertebrates. What's something that's very common among all invertebrates besides the fact that they're invertebrates?
Small.
They're pretty small. Sure, we have some crabs and lobsters that can get pretty big, but on the whole, invertebrates are pretty small. So they can increase the diameters, but it's not going to have a huge impact on their system as a whole. On the other hand, the larger you get, if you're trying to increase the diameters, all of a sudden there's going to be a large proportion of the body mass is going to be associated with the nervous system, which means that you even then have to be larger to support your nervous system. So even though we do have some nerve cells that have extremely large diameter axons, the vertebrates do not use this method of speeding up the signals as much as the invertebrates. The methods that vertebrates primarily use is using what's called a myelin sheath. The myelin sheath are basically a way to insulate the axon to help speed up the signals. The myelin sheath is when you produce this action potential in these cells, and it's moving along the axon. If you have just this axon present, you saw in the animation, you have a continuous stream of positives and negatives moving back and forth. When you have cells that have myelin sheaths, they have specific nodes, specific portions of the axon where this potential can be created, where you can have this switching of the gradient. These are referred to as the nodes of Ranvier. These are specific gaps in the sheath. When you have the sheath there, you can't create a potential, and so the signal travels very quickly through to the next node where the action potential is then continued. So rather than having the signal being produced at every single interval or every single location along the axon, you have it only occurring at these specific nodes, and in between it moves very quickly through. So the impulse sort of jumps from node to node to node to node. And this jumping process helps speed up the signal. So if you look at nerve impulse transmission, this is the axon right here. You can make it a larger diameter to speed it up, or you can have these myelin sheaths where you have these nodes where the action potential can be created. and it will then jump in between each node, speeding up the rate at which the signal is being sent. It'll then be sent quickly on to the end of the axon, picked up by the next cell, and then continued along. Now, eventually it will reach the end of the axon. Once it reaches the end of the axon, the signal doesn't stop there. It needs to be able to make a jump to the next cell. And it does these through what are referred to as synapses. The synapse is basically the intercellular junction between two nerve cells. It's where the axon ends from one cell and where the dendrites begin from the other cell. They're not physically connected to each other. So the signal has to jump across this synapse from one cell to another. Now, in some cases, you have a situation where the dendrites are connected to other neurons. In other cases, you have a situation where the dendrites are connected to sensory cells, or you might have the axon connected to muscle cells or gland cells. So they can be connected to different things. We're going to focus on when it's a neural connection, when you have one neuron cell sending a signal to another one. So with these synapses, you have to have what's called a presynaptic cell, and you have the postsynaptic cell. The presynaptic cell is the one that generates the signal that is going to send it to the postsynaptic cell. The presynaptic cell has to transmit this potential, this action potential, across this synapse. The postsynaptic cell receives it. And now there's two different mechanisms that cells can use to send a signal across the synapse. One of these is with electrical signals, and one of them is with chemical signals. And they're both going to work to send this signal from one cell to the other. The molecules that are involved are referred to as neurotransmitters. And the neurotransmitters basically are there to bridge the gap, bridge this synapse to enable the signal to continue transmission. So we have a quick animation here showing you what's going on with these synapses.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated sodium ion channels to open, and sodium diffuses into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
So basically all we're having is where we have one cell starting to release these neurotransmitters. Those neurotransmitters fill up this synaptic gap and bind to the receptors in the postsynaptic cell. Once they bind, they then open up, allowing sodium ions to flow into the cell. That's going to create a stronger positive gradient inside, starting this process of creating the action potential in the new cells. If it reaches the threshold, then that action potential will start to be sent along to the next cell. So if you think a second, every time something happens in your body and a nerve signal is being transmitted, this process is occurring in between every single neural cell. So, now there's a lot of different kinds of neurotransmitters. They mentioned the acetylcholine. This is one of the ones that's involved. You have neuropeptides. Those are kind of neurotransmitters that are specifically involved with pain reception. So when you're feeling pain, when I told your neighbor just to pinch you the other day, you're going to be releasing those neuropeptides. You have amino acids. There's another, glutamate that can be another neurotransmitter. You have biogenic amines. These are things like epinephrine. This is responsible for the adrenaline response. This is one of the ones we talked about with the fight or flight response. It's a specific neurotransmitter that's going to be responding to a specific kind of signal. You have dopamine that's responsible for body movements. You have serotonin that's responsible for regulation of sleep. So all these different neurotransmitters are responsible for bridging this gap between two different cells depending on the kind of signal or the kind of stimulus that is being sent. It's not just one neurotransmitter for everything. Now, the nervous system themselves, we've sort of given an overview of how the nervous system works, but the actual nervous system varies from animal to animal. The one major phylum of animals that does not have any nerves whatsoever are the sponges. We talked about those as sort of the most ancestral animal. They don't have extreme cell differentiation. They don't have the tissue types. There's no tissue layers or anything like that within the sponges. Every other animal that we're talking about has at least some sort of form of a nervous system. They have some sort of nerve cells that can transmit signals. Cnidarians have the simplest nervous system of all. These are basically just neurons that are linked together to form a nerve net. There's no central area, there's no central nervous system or peripheral nervous system. It's just this nerve net that can respond to stimuli. When we start talking about sort of more advanced nervous systems, so there's no associative activity, they don't have any sort of learning or anything like that. If we talk about more advanced nervous systems, we actually have to start talking about the platyhelminthes, the flatworms. Remember, they're the organisms that don't have a true body cavity, but they do have tissue layers. These are the simplest animals that have any sort of associative behavior. They can start to associate a stimulus with a response. And all that means is that they're capable of learning. So platyhelminthes, with their simple nervous system, are capable of learning behavior, learning responses. They have two nerve cords that run down the body. These permit relatively complex muscle control. It's not just a net of nerve cells, but actually nerve cords that sort of coordinate activity. And so they're sort of the most rudimentary organism that has what we would define as a true nervous system. All nervous systems in every organism can be sort of thought upon as building from this basic system. They take the system that we see in flatworms and add to it to create the nervous systems that we know about. So basically, you all have a more advanced nervous system, but it's still based on this idea of the flatworms. All evolutionary changes in the nervous system, ranging from increase in the parasympathetic or sympathetic nervous system, increase in sensory ability, brain activity, spinal cords, these are all just enhancements of the flatworm nervous system. So here we sort of see some basic nervous systems. We have the cnidarians that have a nerve net. Everything else has some sort of basic radial or some sort of extended nervous system. It's based on this idea of the flatworm one. The humans obviously have one of the more advanced ones. There are certainly other vertebrates that have ones that are just as advanced as humans, however. So let's talk about one of these things that's sort of considered an advancement, the vertebrate brain. All vertebrates have brains, and the brains are broken down into three basic regions. You have the hindbrain, you have the midbrain, and you have the forebrain. The hindbrain is also referred to as a rhombocephalon. The midbrain is referred to as a mesocephalon, and the forebrain is referred to as a prosencephalon. So you have these sort of three different brain regions, and each one of which is responsible for sort of regulating or controlling different aspects of behavior and just different aspects of signal processing. In the vertebrates, you have different regions that are different sizes. The regions of these brains, these three regions, vary as vertebrates have evolved. As they've evolved more specialized nervous systems, more specialized signal interpretation, more specialized behavior, different portions of these three regions have become more advanced, have become larger in size. So in fish, the hindbrain is the largest portion of the brain. If you were to dissect a fish brain and look at it, you would see that the hindbrain is much larger than the midbrain or the forebrain. In mammals, the forebrain is the largest portion of the brain. So we sort of have this continuum where fish, the earliest vertebrates, have the largest hindbrain. Mammals and some birds, among the most recent vertebrates, have a larger forebrain. So we have an evolutionary trend of increasing the forebrain size relative to the other sizes of the regions. So the forebrain in mammals, in many birds, has become sort of this dominant brain feature. If you look at a brain, if you think sort of a human brain, and you think about that wrinkly gray mass, that entire structure is the forebrain. The hindbrain is relatively small, the midbrain is relatively small in mammals as well. So this is a simplified diagram of a vertebrate brain. Doesn't look anything like your brain, does it? But this still has the same three components. This is what we might see in something like a fish brain. You have the hindbrain, which might be the largest portion. The hindbrain is directly going to be connected to the spinal cord. You have the midbrain. Then you have the forebrain, which has the cerebrum, which is where abstract thought takes place. You have the hypothalamus, the pituitary gland, and the thalamus, which are responsible for regulating body activity through releasing hormones. So this is the basic structure of all vertebrate brains. The relative size of these different three sections depends on the kind of vertebrate we're talking about. With the vertebrates that are mammals and humans, the cerebrum has become a very dominant structure, so we see a very large cerebrum. So the forebrain as a whole is much larger than the other three, or the other two sections. Now the increase in the mammals is because of this increase of the cerebrum. One thing we also see is we see a split of the brain occurring. The brain is now split in mammals into the left and the right cerebral hemispheres. So not only do we have a larger cerebrum, but what are we accomplishing by splitting it?
Bilateral symmetry?
Bilateral symmetry. If we just have one large brain managing everything, as opposed to two halves of the brain, what are we increasing?
Specialization.
Specialization. So every time we have specialization, we're increasing efficiency, we're increasing the ability for less effort producing more results. So our split of this brain allows for specialization to take place. They're still connected to each other. We haven't completely split them in two. We need to send signals back and forth. They're connected by the tract that's called the corpus callosum. But each half of the brain, the left or the right hemisphere, is specialized. And in fact, each half of the brain receives sensory information from the opposite side of the body. So your right half of your brain is receiving sensory information from your left side. Your left half of the brain is receiving sensory information from the right side. We also talk about people being left-brained or right-brained, that having to do with whether they're sort of artistic in nature, mathematical in nature, and so forth, because the brain is becoming specialized. Now, the fact that we have this situation where each side of the brain is receiving information from the other side, if you're right-handed, what side of the brain does that mean that you're processing as you're writing? Your left side. If you're left-handed, what side of the brain? Right. So that's why you can really honestly say that left-handed people are the only people in their right minds. Now, the hemispheres themselves are broken into different regions as well. So we start to have specialization not just in the left and the right half of the brain, but also in different portions of the brain that are responsible for different activities. You have the frontal lobes, you have the parietal lobe, temporal lobe, and occipital lobes. These are all responsible for processing different kinds of information. Different parts of the brain are serving different functions within the mammals. If you look at the brain here, these are the different lobes that you can see. Here we have the cerebellum. What was the cerebellum? What portion of the brain? Hindbrain. So you can see that that's connected right to the spinal cord there. But this whole area is the forebrain. So there are areas of the brain that are responsible for controlling speech and motor activity. There are areas of the brain that are responsible for receiving sensory information. There are areas of brain that are responsible for interpreting sensory information. So there's all these different areas that are involved in very specific activities. And you can actually trace different portions of the brain to different activities. And this is one of the areas of research that's being done in neural research. Now, there are other structures in the brain. There's the thalamus. The thalamus is responsible for sort of integrating visual, auditory, and somatosensory information. Somatosensory information just means information from touch. So it sort of integrates all this information to interpret what you're seeing, touching, feeling, smelling. The hypothalamus is responsible for integrating visceral activities, sort of non-sensory information. The hypothalamus also controls the pituitary gland, which is responsible for releasing a lot of hormones. You have the limbic system portion of the brain. This is made up of the hypothalamus, the hippocampus, and the amygdala, and this is responsible for emotional response. So every time you have any sort of emotional response, that's being controlled by the brain. You may think it's irrational, but it's still being controlled by the brain. It's what's being controlled by this limbic system. So you have different portions of the brain that are responsible for different behaviors. Now, there's a lot of brain research that's being done. In fact, just recently there was an article that was being published talking about research into dreams, where they're actually looking and learning how to predict what you're dreaming about based on the activity that's occurring in the brain. They hook up electrodes to someone who's dreaming. They see what regions of the brain are being activated, and they can predict to some degree what you're actually dreaming about. There's actually research being done about predictive thought behavior, where they've done some research saying, okay, you have to choose either this or this. And before you're even consciously aware of making a choice, your brain has already determined which one you're going to choose, and they can identify that with about a 65% accuracy, better than just randomly guessing. So the brain is something that can be mapped, it is something that can be identified in order to understand how thought processes work. It's not just some abstract thing that we can't measure. Brain activity, how we think, how we behave, is something that can be measured. Now, the brain itself is connected to the spinal cord. The spinal cord is basically a cable of neurons that extends from the brain down through the backbone. All vertebrates have backbones. It serves to protect the spinal cord. The spinal cord itself is enclosed by this vertebral column. You also have the meninges that are also there to protect the spinal cord. So the spinal cord you can think of it as the body's information highway. This is where signals are being sent back and forth from the sensory neurons to the spinal cord up to the brain for processing. Once the brain has processed the signal and initiates a response, that's sent back down the spinal cord and back out to the motor neurons where the response is elicited. So all these signals are being sent back and forth through the spinal cord. And so the spinal cord is there to relay the messages between the brain and the body. It consists of these interneurons that just sort of continuously have this signal flow occur. But the spinal cord is also involved in reflex action. What is a reflex action? Involuntary response. You don't have to think about a reflex. That's the kind of thing where if you stick your hand in a fire, you jerk it out before you can actually even think about pulling your hand out. What is the more common reflex response that we always see at the doctor's office? The knee jerk. So the knee jerk is sort of one of these very common reflex responses, and many reflex responses are what we refer to as monosynaptic. That is, there's only one synapse that takes place, one connection between neurons. So where is that synapse going to be? It's going to be in the spinal cord. So you have one nerve cell sending the signal to the spinal cord, and then the signal is not sent to the brain. Instead, it's interpreted directly at the spinal cord, sent around, and the response is sent back. So there's two nerves involved, one synapse that connects them. So there's one single interneuron. So this is sort of what you might see for a reflexive response. You have a stimulus, a sensory neuron that receives the stimulus. It sends the signal up to the spinal cord. It doesn't get sent to the brain for processing. Instead, it's processed right here and immediately sends a response to the motor neuron where the response is initiated. So that's referred to as a monosynaptic reflex. Monosynaptic because there's only one synapse. Reflexes, by definition, are going to be very quick. They're going to be involuntary. Usually reflexes are going to be behavioral responses that have to do with something that is going to be extremely threatening, extremely important to respond to very quickly. Now, knee-jerk response is not one of those, but if you think about some other reflexes where something happens and you don't even think about it, usually it has to do with pulling your hand away with pain, with heat, or something along those lines, where you don't have time to think about or process what you want to do. So that was sort of the central nervous system. We then also have the peripheral nervous system. The peripheral nervous system are these nerves and ganglia that are receiving information from the environment and then sending signals to the specific motors, to the specific structures that are going to respond. So you have the same kind of nerve system. You still have the nerves with the nerve cell body, with the dendrites, with the axons, receiving signals, creating this rated or action potential, sending that signal to the other nerves. But with the peripheral nervous system, you don't have any of the processing. That is, it's just picking up the signal from wherever it's originating, either from the senses or from the brain, and then conveying it somewhere else. Either conveying it to the central nervous system or away from the central nervous system. So the peripheral nervous system is not comprised of interneurons. It is comprised of either sensory neurons that are picking up the information or motor neurons which are receiving a signal. The sensory neurons, when you look at those, the axons enter the dorsal, axons from the sensory neurons enter the spinal cord. So they enter the dorsal side. Every single sensory neuron that sends a signal to the spinal cord, it enters the dorsal side. This forms the dorsal surface, the dorsal root of the spinal nerve, the spinal cord itself. Cell bodies of the sensory neurons are usually grouped around the spinal cord on the in what's called the dorsal root ganglia. So it sort of helps to help speed up the processing of signals. The motor neurons, you have the axons leaving the ventral surface, leaving the spinal cord. It leaves from the ventral side, so the dorsal side is where signals are sent in. The ventral side is where signals are sent out. This forms the ventral root of the spinal nerve, and the cell bodies of these motor neurons that send signals out are actually located in the spinal cord itself. So they have the cell body in the spinal cord, and then the axons extend out from the spinal cord. Now, the peripheral nervous system is divided into the somatic and the autonomic nervous system. The somatic nervous system is responsible for muscle contraction. These are things we can control. We can decide to bend our arms, to start doing jumping jacks, to wiggle our ears, whatever we want to do. Well, some of us do. But what that is under conscious control. That is part of the somatic nervous system. So the somatic nervous system is going to be anytime you have a conscious command, but it also involves reflex action. Those monosynaptic reflexes are part of the somatic nervous system. Why is that? It's because those neurons are connected to muscles that we normally can control. We normally can control our leg to bend. But when we have a reflex response, our leg bends without thinking about it. It's a reflexive action. But it still is the same muscle group. It's still the same nerves that are involved. The autonomic nervous system is one that's not under our control at all. And it's broken down into sympathetic and parasympathetic nervous systems. So we have the sympathetic nervous system, which we aren't under conscious control, and the parasympathetic nervous system. It's also comprised of the medulla oblongata that helps sort of regulate this activity. So the parasympathetic is not, or the autonomic is not under conscious control. When we talked initially about the parasympathetic and the sympathetic, I mentioned that one of these is involved in fight or flight responses, and one of these is involved in rest or repose responses. Here's a description of what different systems are under sympathetic or parasympathetic control. Which one of these is fight or flight, do you think, and which one of these is rest or repose? Look at what these responses actually are.
Sympathetic is fight or flight.
The sympathetic is fight or flight. How many people agree with that? It is. Look at what's happening with these responses. We have the speed up the heart rate is under responsible of the sympathetic. If you're engaged in an activity where you're fighting, fleeing, fighting, something like that, you want to speed up the heart rate. You're going to secrete adrenaline. That's under the sympathetic nervous system. Your pupils dilate. That's under sympathetic nervous system. So these are all activities that are under sympathetic nervous system control. Peripheral nervous system responsible for fight or flight. On the other hand, you have the parasympathetic that are responsible for sort of the rest and repose responses. Things like increasing, slowing down the heartbeat, constricting the bronchioles, constricting the eyes, secreting saliva, increasing motility. These are all things that are referred to as rest or repose behaviors, sort of slowing the body down. The sympathetic you can sort of think of it as speeding the body up. The rest and repose is slowing the body down. It doesn't mean that the parasympathetic controls when you go to sleep, but certainly some of the things that occur when you sleep or when you're trying to go to sleep are under control of the parasympathetic nervous system. So that brings us to the end of the nervous system. So the next chapter we have, this will not be on test number 3, this will be on test number 4 now, is the digestive system, chapter 48. Now, when we talk about digestive systems, we're talking about heterotrophic organisms that are absorbing ingesting food and absorbing the nutrients from that food. We're not talking about the autotrophs that can produce their own food. So with the heterotrophs, the kind of food, the way that they get their food, they're classified based in three different ways. The three different categories of heterotrophs are based on what their food sources are, where they're getting these nutrients. We have some that are referred to as herbivores. These are animals that exclusively eat plants. So they get all the nutrients they need from plant material. You have carnivores. These are animals that exclusively eat other animals. They get everything they need from their other animal tissue. And then you have some that are omnivores. These are animals that will eat both plant and animal matter. In some cases, it will be varying based on availability. In some cases, it will be varying based on behavioral preferences. But they're capable of eating both. Which ones are humans? Omnivores. Don't your parents always tell you to eat your vegetables? You would much prefer to have a cheeseburger or something like that? So you're an omnivore. You can have both the animal and the meat tissue. A lot of animals that we don't think of as omnivores actually are. Bears, for example, are omnivores. They actually eat much more plant material than they do of animal material. Dogs are omnivores. Under natural conditions, they eat more plant. They eat a lot more plant than we normally give them. But they can eat the plant material. They can eat the animal material. Animals that are exclusively herbivores are going to have digestive systems that are designed to break down only plant material. Animals that are exclusively carnivores are going to have digestive systems that are designed to break down only animal material. But the omnivores need to have sort of a jack-of-all-trades digestive system. They need to be able to break down animal material. They need to be able to break down plant material. Now, when we talk about digestive systems, the simplest kind of digestive system would be that we see in a single-celled organism. These organisms and the sponges don't have what we would define as a digestive system. What they do is they break down food extracellularly and then absorb those nutrients. Multicellular animals are what we define as having a true digestive system. They usually have some sort of body cavity where food digestion takes place, not just outside the cell itself, but rather within the organism, but outside the cells. Single-celled animals can't do this. Single-celled organisms can't do this because they're single-celled. They don't have any sort of body where they can have this body cavity. So with the multicellular organisms, we're looking at what we refer to as a digestive cavity. This is the structure where digestion takes place. Now, cnidarians and flatworms have a very simple gastrovascular cavity. They have a very simple digestive cavity where digestion takes place. There's only one opening, and there's no specialized regions. Why do we need specialized regions? Well, we need it for digestion, but they don't. Well, why do we need specialized regions? We meaning more advanced animals, not just us, humans.
To break down more specific minerals?
To be more efficient. So when we have specialization, we're talking about an increase in efficiency. We can talk about breaking down more types of food matter. So we can have a specialization where we can have an area that breaks down proteins. We can have an area that breaks down lipids. If you have a simple gastrovascular cavity with just one opening, there's no specialization. Anything that goes in is going to be exposed to the same digestive process, and any waste materials that could be excreted through that same opening. So specialization occurs when you have a digestive tract. Starting off, the first form of specialization is having two openings. You have an opening in the mouth region and an anus opening. So you have one opening that's specialized for receiving food, one opening that's specialized for excreting wastes. That is a form of specialization right there. Now, nematodes have the simplest organism that have this form of specialization. When we have this form of specialization, two different openings, we refer to it as a digestive tract. Because the food is going to enter one end, it moves through this tract, and is processed, and then leaves the other end. So nematodes have the most primitive of all digestive tracts. They have these two openings, but they don't have much specialization beyond that. Now, when you have this digestive tract, remember, all systems in the animal bodies are lined by epithelial tissues. If you have tissue types, you have epithelial tissue types. So the digestive tract, the gut itself, is going to be lined with epithelial tissues. If you don't have tissue types, then you cannot have a digestive tract. As we see more advanced animals that have more specialized dietary needs, we see more advanced digestive tracts. The digestive tracts themselves become specialized into different regions. So you can have a more efficient process of extracting nutrients, extracting energy, extracting minerals, whatever you might need from the food material. So pull out your clickers. We'll end with this question. Actually, we won't end with this question because I answered it already. The evolution of the separate openings for the mouth and anus leads to specialization within the digestive tract. Why is that? Look at these answers. Is it A, the mouth and anus allow for directional flow of food? B, the mouth and anus evolved into a specialized digestive stomach? Or C, the mouth and anus each secrete specialized digestive juices? B. How many people think it's A? Raise your hand.
Not even know.
How many people think it's B? Raise your hand. How many people think it's C? Raise your hand. So B is the most common one. It is wrong. Good thing we didn't have this as a question. The idea with this digestive tract is there's a directional flow. Okay? Now, keep it down there. Come on. Food flows in one region. Once it flows in, you have a directional flow. It's only going to move in one direction. So you can have a continuous processing that takes place. It will eventually enter the stomach. The mouth did not evolve into a stomach. The anus did not evolve into a stomach. But instead, part of this digestive tract evolved into what we would refer to as the stomach. So the presence of the mouth and the anus allow for specialization because it's always going to move in one direction. You always have processing that's going to take place in the same order. You don't have to worry about food coming back up the same way it came in, where you can't have specialization because you're going to continue to see different levels of food processing. So A is the correct answer here. We'll start talking about different kinds of digestive systems on Wednesday.
My personal problem is I took this morning.